Let me see. Swap these. Can you see just my slides here? All right, cool. So yeah, thanks for having me. And thanks again for giving me kind of this opportunity to present some of my past research and how I'm going to take that and apply it to the research program I'm just now starting here at UH Downtown. So first, what I'm going to do today is kind of provide a brief background of my research interests in animal behavior and how they kind of led me to the housefly study system. I'll then talk about kind of the three main focuses of what will be my research program, involving the roles of genetics, ecology, and chemical communication as it relates to animal behavior. And then throughout the talk, I'll kind of give some project ideas we have regarding where we plan to take research in this system in my lab. So my main interests lie in determining the genetic and environmental mechanisms by which interesting behaviors are developed, with a particular interest in the development of different social and mating dynamics, both within as well as among species. It's a topic that's interested me since I started conducting research in undergrad, when I first kind of got interested and exposed to animal behavior, where I was studying predation in damselfly larvae, this species here over at Rice University. I then, for grad school, switched gears and looked at female mating preferences in swordtail fish, and then, have worked in a number of insect systems, and am now kind of focusing on the housefly. And to address these behavioral questions that I'm interested in, my research typically applies a multidisciplinary approach, where I draw on principles and methods ranging from chemical and thermal ecology to genetics, and ultimately coupling these skills to address questions related to animal behavior. And this sort of integrative background really reflects my research history. So each position I've kind of held throughout, my scientific career has sort of allowed me to develop new methods and apply them to research topics in animal behavior. And so specifically, my research is focused on this topic of how genetics and the environment interact to affect the development of ecologically and socially relevant behaviors. So first of all, what do I mean by ecologically relevant behaviors? These pertain to those behaviors that are either influenced by or in turn influence important ecological processes. So we can think of things like predatory behaviors, where habitat structure can affect the different strategies that predators will use to catch prey. This can relate to thermal preference, where an individual's genetic background might affect how individuals within a population occupy space within their thermal habitat. And then it can also be things like colonization or oviposition preferences, where the microbial community on a resource of interest affects its chemical signature that many animals use to locate that resource. So these are all examples of just some ecologically relevant behaviors. We contrast this with social behaviors, which focus on behaviors that are either influenced by or, again, influence other members of the same population. So things like female or male mating preferences, as well as personality-related traits, like how aggressive an individual is, or how shy or bold they might be within the population. These all influence how an individual interacts with or responds to a member or members of their own species. And one of the more interesting systems that serves as a nice sort of intersect between studying ecological as well as social behavior is actually the housefly. Now, the housefly has a lot of things going for it that make it particularly attractive to studying both basic and applied research. So on the applied side of things, the housefly, as well as muskid flies in general, so these are members of the family Muscidae, things like stable flies and horn flies as well, they can cost billions of dollars worth of damage to livestock and other agricultural businesses worldwide. They can often be such terrible pests to things like livestock animals that it causes things like extreme stress, impaired immune responses, and ultimately weight loss in these animals. They're also potential vectors of lots of nasty diseases like salmonella and weight loss, salmonella and tuberculosis, And it was even recently shown actually that, it's just a preprint right now, but they might be able to harbor COVID as well. But I think the jury's still out on that one. So there's a vested economic as well as public health interest in studying the behavior and ecology of this insect in order to, most importantly, control their populations. Now, one of the most effective means of controlling housefly populations is a nice sort of proof of concept I like to bring up on how studying animal behavior can have a real applied benefit. In many livestock facilities, you'll often see these olfactory lure traps used to control housefly populations. And these traps are laced with housefly female sex pheromone, muscalure, or Z9 trichosine. And so by just studying this system from a basic animal behavior perspective, we can see that an applied benefit was gained that's been effectively used to control populations of these pests. Now, from a basic science perspective, houseflies have pretty interesting social behaviors. And there's a wealth of older literature that kind of describes male housefly mating behavior with a focus on specifically what components of the female pheromone profile elicit male attraction or aversion. Basically, candidate compounds like muscalure would be identified using gas chromatography and mass spectrometry in order to identify candidate female pheromones. And then male response to these chemical compounds would be assayed in a number of ways. So here I'm just showing you an example of a Y maze where a fly here is introduced to the bottom, and you see if the fly goes to the control chamber or the chamber housing the odorant of choice as a sort of measure of their relative preference for that candidate compound. But even though there's a lot of older literature out there, there are plenty of gaps in our knowledge of social behavior in the housefly system. For one, there's almost no research whatsoever on the female side of mating behavior. And this is particularly baffling to me because this is a system where theory would predict that female behavior is particularly important. Specifically, unlike fruit flies, female houseflies will typically only mate once. And theory suggests that in these systems where females mate only once, and there's a great degree of sexual dimorphism, and where reproductive costs are higher for females, then female mate choice would be predicted to play an important role, as it pays more for the female to be choosy in who she mates with. And so this seems to define the housefly system, but for whatever reason, female mate choice and mating preferences have rarely been studied at all in this system. And so this huge gap creates a lot of low-hanging fruit projects that are particularly amenable to undergrad research, which is the type of institution I've recently been employed at, a primarily undergrad institution. Houseflies are also a particularly unique system to work with regards to studying the evolution of sex determination pathways and sex chromosomes. So first of all, sex determination is just the process by which genetic differences or environmental cues initiate the development of these sexually dimorphic traits that we so often see between male and female across animal systems. And so one other thing is that evolutionary transitions in the sex determination pathway are often accompanied by this rapid turnover of the sex chromosomes, which can explain why sex chromosomes are often non-homologous across even closely related species. So what do I mean by that? So just for example, in this family of rice fish, you can see that there's rapid evolution ultimately of which chromosome ends up becoming the sex chromosome for any given species. So these are all the members of the same genus, but as you can see, the sex determining chromosome varies from species to species. And so because of this, a major goal in evolutionary biology is to identify the factors that drive the evolution of sex determination pathways, as well as just identify what the consequences of developing new sex chromosomes are. So houseflies are pretty unique in this aspect because they exhibit what's called polygenic sex determination, where the primary male determining gene can be found on any of the 6 chromosomes that houseflies have. And so they're not just restricted to having any single chromosome designated as its sex chromosome, like you see in most species, including humans. And so because of this, in males, we refer to chromosomes carrying that male determining gene, MDMD, as that male's proto-Y chromosome. MDMD here just stands for Musca domestica male determiner. That's what that stands for. But yeah, wherever this gene is located, that chromosome becomes that male's proto-Y chromosome. Now, while this gene can be found on almost any chromosome, in nature, we find it most frequently on what was originally the ancestral Y chromosome or on the autosomal third chromosome. And I'll refer to these as YM or 3M male types throughout the talk. So in nature, we find that polygenic sex determination has been maintained as this stable polymorphism between 3M and YM male types for at least over a century. YM and 3M males are distributed along latitudinal clines across multiple continents. Here I'm just showing you the eastern US, but this has been shown in Japan as well as Europe, where we find this same pattern where 3M are most common in the southern warmer climates, and the YM type it prevails in the northern cooler climates. And so as you might expect, temperature has been shown to be the best predictor of the 3M and YM frequencies across populations, but this hasn't really been empirically tested until recently in work that I've done in the MISO lab. And a little bit later, I'll talk about some results we've gained with regards to the role of thermal ecology in this system. But what I want to shed light on first is this interesting difference in mating behavior between these 3M and YM male types. Specifically, in the lab, it's been shown that 3M males will outcompete YM males in competitive mating assays. So specifically, if you give a female the option to mate with either a YM or a 3M male type, more often than not, she'll mate with that 3M male type. In multi-generational cage assays, this mating advantage translates to the rapid extinction of the YM genotype. And so this leads us to ask, why do we see maintenance of genetic variants of this male mating success in nature? And this is a nice segue into kind of the core research interests of my lab, which are identifying the genetic, ecological, and chemical mechanisms that drive the development of ecological and social behaviors in the system. And so diving into that first topic, which aims to understand the genetic architecture of social behaviors like male mating behavior. And I'll talk about two aspects of this research with regards to two families of housefly strains that I'm incorporating in my research program. And those are the CS and Abies families of housefly strains. So I'll be using the CS background strains to determine the effects specifically of sex chromosome evolution on behavior, whereas the Abies family of strains will be used to identify autosomal chromosomes that are important in regulating behavior. So sex chromosome versus autosome. So first of all, regarding these CS strains and sex chromosome evolution, we can use strains developed from a common genetic background to ask what effects that specifically the male proto-Y chromosomes have on male mating success, and how might variation in these traits be maintained in the wild. And the way we develop these strains is pretty neat and straightforward, with little to no genetic sequencing required. So in housefly, we have this nice system set up where we basically start with one strain, the CS strain, which is historically a 3M strain, the M here referring to that male determining gene. And through a series of back crosses, we create multiple replicates of 3M and YM nearly isogenic lines that only differ at the chromosomes in which the male determining gene is located. Everywhere else, they're essentially genetically similar. Females, what that means is that females, which of course lack the male determining chromosome in this system, are all genetically similar across strains. And so because of this, when we compare things like RNA-seq or behavioral data between these 3M and YM strains in our lab, we're essentially asking what the effect of just this male determining chromosome is on our trait of interest. In other words, we're controlling for effects caused by differences in the genetic background, something you wouldn't be able to account for if you just took 3M and YM males directly from the wild. And so one of the first things I did when I joined Dr. Meisel's lab was I just wanted to compile all the RNA-seq data there was. And I conducted this meta-analysis on all the RNA-seq data there were from the head tissue of 3M and YM males that were derived from these nearly isogenic CS background strains. I conducted my own gene expression and network analyses on this pooled expression data set to see what interesting mating related genes pop up as differentially expressed consistently between 3M and YM males. Now, in general, there were only a handful of differentially expressed genes at all, but one that stood out was the only mating related gene within that list, OBP56H, which specifically is downregulated in 3M males. It's one of a family of odorant binding proteins, which are typically involved in the detection of odorant cues, but they have other functions as well. And this one in particular caught my eye for a couple of reasons. First of all, in a previous study in Drosophila melanogaster, it was shown that suppression of the expression of this gene actually has this drastic behavioral effect. Specifically, males, but not females, with suppressed expression of this gene were faster at mating. In other words, they had lower copulation latencies, which provides those males with a mating advantage. Similarly, in our houseflies, 3M males have lowered expression of OBP56H, and they also have a mating advantage by outcompeting YM males for mates. Now, of course, this isn't the exact same as what we saw in the Drosophila melanogaster, since this housefly assay isn't directly testing the speed at which mating was initiated, but it does seem to suggest that there might be this conserved behavioral function of this gene that's conserved between Drosophila melanogaster and housefly. But we weren't satisfied. We needed some behavioral trials of our own to really solidify this connection. And so we conducted our own behavior trials on housefly, testing a couple different things. First, we asked, is this mating advantage for 3M males temperature sensitive? So remember, temperature is a good predictor of these genotypes in nature. So perhaps there's an important genotype by environmental interaction going on that might explain how this difference in mating behavior is maintained. The previous mating results I showed were conducted only at a warm temperature. This is warm for houseflies, 28 degrees. And this temperature we would predict benefits 3M males based on the fact that they're found at these lower latitudes where it's warmer. So then perhaps, YM males might outcompete 3M males if we do the same test at a colder temperature. And so the way we did these trials is pretty straightforward. We took flies reared for multiple generations at either cold or warm temperatures. And then we take females from a genetically unrelated strain, and we put her in an arena with a 3M and a YM male. We provide them all with food and water, and we just see what percentage of females mate with 3M males over YM males in a given trial. When we replicate this over several batches, what we found was that this 3M male mating advantage seems to be independent of rearing temperature, at least based on the temperature range we tested, which is pretty low for houseflies. We can't really raise them in our lab lower than 18 degrees. And so we see that at both cold and warm temperatures, females mated with a significantly greater proportion of 3M males, where the red line here is just marking that 50-50 null expectation. And so by conducting these trials, we were able to kind of rule out the possibility that a genotype by environment interaction involving environmental temperature explains the maintenance of these two male types in nature. Now, with some undergrads, we were able to find some interesting temperature effects on different mating behaviors, but I can talk about that more later if you're interested. But for the sake of this talk, the second behavioral question we wanted to address was whether this 3M male mating advantage is it driven by differences in copulation latency between male types? In other words, we wanted to replicate the design used in that fruit fly study, which showed that OBP56H suppression results in male fruit flies mating more quickly. And so if we get a similar result in housefly, then this would suggest that maybe there is a genetic pathway linking OBP56H expression and male copulation latency, a genetic pathway that's conserved between these two species. And so this time we conducted just single choice assays, similar to those done in the fruit fly experiment, with the hypothesis that 3M males, which again have reduced OBP56H expression, 3M males should mate more quickly than YM males. So specifically, we asked, if we conduct these single choice assays, do we see a greater proportion of 3M males mating within the first 4 hours of a trial? And that's exactly what we found. During the first 4 hours of a mating assay, about twice as many 3M males had already mated than compared to the YM males. And this pattern seems to hold regardless of the cutoff time we used, where we consistently see a smaller proportion of unmated 3M males than YM males. So this all seems to support this idea that the idea of this link, that this link between OBP56H expression and male mating behavior is conserved between our houseflies and fruit flies. So these behavior trials were, I just want to note that these were largely conducted by undergrads in the lab using methods that are fairly easy to get accustomed to. And so these are the types of assays that I'm going to kind of continue doing at this primarily undergrad institution, testing other behaviors as well, seeing if aggression differs between male types, etc. We after conducting these behavior trials, we might also be able to couple these behavior results with data obtained from other genetic or molecular studies. And here I'm not talking about conducting costly sequencing experiments. Rather, we can use what's already out there, like I already used in my gene expression analysis. So for example, looking back at OBP56H, we used publicly available sequencing data to conduct our own new genetic analyses. Specifically, we wanted to know, what is OBP56H actually doing? In other words, what genes or pathways are regulating or regulated by OBP56H? And to get to that, we can first look at the genetic architecture of houseflies and fruit flies. So the chromosomal structures of housefly and fruit fly are actually fairly similar, and we can order them by what's referred to as molar elements in order to identify homologous chromosomes across species. So for example, the housefly chromosome 3 is homologous to the Drosophila X chromosome, and we refer to this all as molar element A. So OBP56H in both Drosophila melanogaster and housefly are found on molar element C. So in Drosophila, this corresponds to the single copy of the gene that's found on the right arm of chromosome 2, whereas houseflies actually have multiple copies of this gene on chromosome 5. And in case you're wondering, most of these copies of the gene exhibit the same expression pattern. Sorry if I don't show this figure too long for the sake of time, but basically we found that a good proportion of these different copies of OBP56H are actually behaving similarly, with lower overall expression in 3M males in all but just a couple of cases. Now, because the houseflies we tested share a genetic background that's derived from that original CS strain I mentioned before, then we know that their OBP56H genes should all be similar in terms of genetic sequence, in terms of comparing 3M and YM genetic sequences. So this would suggest instead that OBP56H expression might be regulated by some chromosome 3 or some chromosome Y genes, so molar elements A and F respectively, since those are the only parts of the genome that actually genetically differ across the male types we're studying. And so we can actually identify genes that are co-regulated with OBP56H genes by conducting what's called weighted gene co-expression network analysis. So we use this network analysis to identify important modules or clusters of highly covariant genes that differentiate between 3M and YM male types. We then determine whether OBP56H genes get assigned to any of these modules, and then specifically look at what genes are most strongly co-regulated with OBP56H genes on average, in the hopes of kind of revealing what the genetic architecture of these genes regulation pattern look like, and how that might be implicated in affecting mating performance between male types. So when we did WGCNA, we found just one small module that's upregulated on average in 3M males. And this is just visualizing that module. It consists of about 122 genes. And so when we ask what the hub genes are, in other words, what the genes that might be driving the expression of this module are as a whole, what we can do is rank genes according to how strongly a given gene is correlated on average to every other gene within that module, what's called its intramodular connectivity, or K within. And when we rank these genes according to K within, we see that there are really 5 genes that separate themselves from the rest of the genes in the module listed here. So OBP56H is not one of these hub genes. But what we do see is that 3 of these genes, the ones bolded here, are found on housefly chromosome 3, while the chromosomal location of these other 2 genes remains unknown due to not having, the best annotation on our housefly genome. And one interesting side note about these chromosome 3 hub genes that I'll just flash up here is that all 3 show some level of allele biased expression for the 3M allele as well, suggesting that these genes might make for strong candidates in the future. Now, when we look at whether there are any OBP56H genes at all in this module, we do find that this module is actually significantly enriched for copies of OBP56H-like genes. Specifically, 3 of the 9 OBP56H genes are within this small module. Using WGCNA, we can test our hypothesis that OBP56H genes are perhaps strongly tied to chromosome 3 genes by asking if the genes that are most strongly correlated to OBP56H gene expression are disproportionately found on chromosome 3. And regardless of the cutoff we used, we found this sort of overwhelming result that the genes that are most strongly correlated with OBP56H genes on average are disproportionately found on chromosome 3. And so these correlational data suggest that there's this connection across chromosomes between OBP56H on molar element C and molar element A on housefly. However, because these houseflies that we tested genetically differ only at the 3M and YM chromosomes, we can infer that at least some of this connection represents genes on molar element A regulating OBP56H expression. Of course, this doesn't rule out that OBP56H itself doesn't also in turn regulate some chromosome 3 genes. And we can actually examine this by looking back at fruit flies. So in that same OBP56H expression study in fruit fly, the authors also collected RNA-seq data on male head tissue, just like we did with housefly. And so if OBP56H genes in turn regulate molar element A genes, then we would expect that OBP56H suppression in fruit fly results in the disproportionate differential expression of molar element A genes. And that's exactly what we found. What's more is that this enrichment appears to be male specific, since we don't observe this enrichment of molar element A genes in females. And so these results from Drosophila melanogaster provide for us some confidence of this directional connection from OBP56H on molar element C back to molar element A. Now, we also have more evidence of this conservation, including specific genes that are similarly co-regulated in housefly and in fruit fly. I don't really have time to go into it, but can talk more about it later. But coming back to our question of how genetic variance for a sexually selected trait like copulation latency is maintained in nature. overall, our results seem to suggest that there's this general evolutionarily conserved trans-regulatory loop between OBP56H on molar element C and genes found on molar element A. This complex regulatory loop across chromosomes could at least in part explain why we end up seeing genetic variants in male mating success preserved in the housefly system. So by using a network analysis and allele-specific expression measurements on previously published RNA-seq data from both housefly and fruit fly, we were able to find evidence not only that the housefly 3M chromosome might be this trans-regulator of OBP56H gene expression, but that this trans-regulatory loop that affects male mating behavior is conserved in both fruit flies and houseflies. It's perhaps because of this conserved and complex regulatory network that variation in male copulation latency is ultimately maintained. Now, this has been a large collaborative effort that included a lot of undergrad help, and the results from these behavioral and genetic analyses are now being or under revisions for hopefully acceptance at eLife, but we'll see. And so I present these genetic results as an example of how we can use data sets that are already out there and gain new information at virtually no cost. Remember, these analyses are all new findings, but taken from free previously published data sets. And it's these cost-effective yet informative analyses that are going to be some of the types of analyses I intend to keep applying throughout my career. So all the research I've shown so far has involved the CS family of housefly strains, which were generated to determine the role specifically of sex chromosomes in shaping behavior. And actually, for the sake of time, I'm going to kind of breeze through this portion, which is mostly just describing a strain where we'll kind of complement the sex chromosome evolution research to understand how autosomes, different combinations of autosomes might regulate different phenotypes. For the sake of time, this is just a small section, but it mostly serves as a segue into the next pillar of research, which involves thermal ecology. Specifically, I'm interested in determining how ecology, specifically thermal ecology, can play a role in affecting mate choice behavioral dynamics. Now, this idea linking ecology and sexual selection comes from lots of research where I and a large group of undergrads, again, in the MISO lab, conducted studies where we compared thermoregulatory traits of males and females from these CS background 3M and YM strains. Now, so everything I'm showing here was recently published in Evolution Letters, and I believe that's the article I chose for the class portion of this seminar. I might be wrong though, sorry about that. So remember that environmental temperature seems to be the best factor in predicting the distribution of these 3M and YM chromosomes in nature, with 3M occurring mostly in warmer environments and YM more common in cooler ones. So one thing we were interested in the MISO lab in seeing was how the 3M and YM male chromosomes affect their thermoregulation. And this is work that looks again at those CS background housefly strains. We found that the male sex chromosome alone actually affects a few important thermoregulatory traits. So specifically, it affects the temperature sensitive response that males have with regards to heat and cold tolerance, as well as what I'll show in a second, their thermal preferences. So what I mean by this is that for thermal tolerance, we find this predicted response of greater heat tolerance in the warm adapted 3M males and greater cold tolerance in the cold adapted YM males, but these responses depend on developmental temperature. So specifically, 3M males have better heat tolerance only if they themselves are raised in heat. And then the same is true vice versa. We see this genetic difference with YM males having greater cold tolerance only in this consistently cold environment. So it goes away if you switch the temperatures that they're raised at. So this result seems to point at this interesting interaction between male genotype and developmental temperature in terms of shaping thermoregulation. And so this might actually be the G by E interaction that explains how these two male proto-Y chromosomes might be maintained in nature. So we thought this was pretty cool. And with regards to behavioral thermoregulation, we find something sort of similar. Specifically, 3M males prefer warmer temperatures, as we might expect, and YM males prefer colder temperatures. But again, it's dependent on developmental temperature. We only see this difference when they're raised at the right temperature. If you raise them too hot or too cold, then both types of males will navigate to a temperature that sort of compensates for those extreme temperatures they're raised at. Meanwhile, females reared at the intermediate temperature did not differ in thermal preference across 3M and YM strains. And remember, this makes sense because since the strains we tested only differ genetically at the level of the male determining chromosome, then females across strains are all genetically similar and shouldn't differ in those traits. And so this was a nice sort of control result we got, but I'll come back to this. And the way we tested this was by using a thermal preference gradient that I used. It's extremely straightforward with some lanes to kind of keep flies from interacting with one another, all on top of an aluminum slab, with one end dipped in hot water and the other end dipped in ice. This creates a gradient of temperature, in our case ranging from 18 to 40 degrees Celsius. And if it's set up well, you can see individuals start to cluster near their preferred temperatures over time and estimate what that temperature actually is with some temperature sensors placed at known locations. Now, how does thermal preference, though, tie into mate choice? Remember, this research pillar is about the interplay between thermal ecology and social behavior. Well, things get interesting when we look at the thermal preference of females. So even though we didn't really find any difference between females from the 3M or YM strains, something interesting does appear when we zoom in again on this intermediate temperature of 22 degrees. Specifically, females, here shown in yellow, show a bimodal distribution of thermal preferences, with some females preferring temperatures that align perfectly with the mean thermal preference of YM males, and other females showing preferences that match with the peak thermal preference of 3M males. And we know this isn't a result of whether the female came from a 3M or YM strain, because when we break this up according to whether the female we tested came from a strain that had 3M or YM males, we get the same bimodal distribution. And so what this suggests is that if we imagine a sort of natural environment where there are some warm and some cold pockets, then 3M and YM males are going to get access to different types of females. Specifically, they'll get access to warm and cold preferring females, respectively. Now, there's another thermal behavior we can look at. Instead of just looking at the average temperature that a male or female preferred, we can instead ask what the range of temperatures different types of males and females occupy along our gradient, a measure often referred to as the thermal breadth, where a higher thermal breadth signifies a greater range of temperatures that an individual will occupy. And when we compare thermal breadth values, we find that the females that prefer colder temperatures, that's the FC label here, C is just for cold, they occupy a significantly higher range of temperatures than warm preferring females and than both YM and 3M males on average. So what does this mean in terms of housefly mating dynamics? Well, this means that at the level of a local habitat, YM males are pretty much only seeing these cold preferring females on account of YM males having low thermal preference and a narrow range of temperatures that they're willing to occupy. 3M males, though, they get sole access to warm preferring females in addition to potentially access to the cold preferring females on account of cold preferring females having a much wider range of temperatures that they'll willingly navigate. So this could then have major implications on mating dynamics and ultimately the population structure of these flies in nature. And so moving on with this and where my research program will kind of go from here, there are a lot of these small scale behavioral projects that I plan to conduct. First, we can ask, do cold preferring and warm preferring females differ in who they choose to mate with? Is there perhaps some sort of assortative mating going on, where where cold preferring females mate more like, or are more likely to mate with cold preferring YM males, and vice versa with the warm preferring females. And we can test this using competitive mate choice assays similar to the ones I've shown before. We can also flip this and ask, does female thermal preference affect their own attractiveness to males? Now, temperature affects female sex pheromone development in housefly, so it's certainly possible. And we can get at this through conducting male mate choice trials. Now, further down the line, we could combine behavioral assays with actual field work. So I'm pursuing external funding from Texas Ecolab to ask whether thermal preference of wild-caught female houseflies predicts who these females mate with in the wild. But basically, by conducting these types of studies, we can gain a better understanding of how thermal ecology can affect not only an ecologically relevant behavior like thermal preference, but how it can also indirectly influence mating dynamics in nature. Now, for the last couple minutes, I'll talk about chemical communication. Now, while the first two pillars of my research program investigate the genetic and ecological mechanisms that shape behavior, this last one focuses on identifying the chemical mechanisms and candidate compounds involved in many of these important behaviors. So in insects, chemical signaling can be classified into two groups differing in their range. So just basically, there can be long-range communication, which is usually mediated by the production and detection of these highly volatile organic compounds, or VOCs. And we contrast this with short-range or even contact communication. Now, this involves chemical communication at very close proximities. And it's most characteristically involves the production and detection of cuticular hydrocarbons, or CHCs, which are just long carbon chains of varying length found on the exoskeleton of insects. The female housefly pheromone, muscalore, that I showed earlier in the talk, is one example of just a single CHC. These CHCs, as you can see, they can differ in chemical composition. They can contain methyl branches or double bonds, which ultimately influence their chemical properties as they relate to their two primary functions, which are desiccation resistance and social or mating communication. In other words, CHCs help to keep the individual as hydrated as possible, and they also serve as important mating cues to members of the same or opposite sex, depending on the compound. So cuticular hydrocarbons serve as this really cool trait that's under both sexual and ecological selection, two selective pressures that are often at odds. Now, during my first postdoc at Texas A&M, before my time in the MISO lab with Dr. Jeff Tomberlin, and in collaboration with Angel Helms there, I learned how to extract, assay, and analyze chemical signaling data from a variety of insects. I typically conducted these assays to address questions focused a little bit on animal behavior, but mostly for forensic entomology purposes. But currently, I'm using these methods and collaborations to identify how cuticular hydrocarbon profiles differ between 3M and YM males, where we found some interesting preliminary data already, where basically this is not an NMDS graph, where space here is really the important thing to look at, where closely spaced dots suggest more similar cuticular hydrocarbon profiles on average. And basically what we're seeing is that our 3M strains might actually smell more like females than they do smell like YM males. Now, this is ongoing work in collaboration with one of my current undergrads and Angel Helms at A&M. And this is research I'll continue to pursue here at UHD. Specifically, we're going to have undergrads identify candidate compounds that differ consistently between 3M and YM males. Here you can see we already have one candidate compound based on this preliminary analysis. It's just this 29 carbon chain methyl branched cuticular hydrocarbon. But what's interesting about it is it seems to be almost completely unexpressed in YM males, but fully expressed in 3M males as well as females. And so what we'll do from here is conduct different behavior trials to see how males or dummies laced with these compounds affect their attractiveness to females. And so by conducting these trials, we'll gain a better understanding of the chemical compounds involved in regulating mate choice. So basically, we'll take a blank cuticular hydrocarbon housefly profile, perhaps a YM cuticular hydrocarbon profile, and then compare it to one that's laced with our candidate compound and see if this affects their overall attractiveness. We can also follow up these studies with further studies that test their implications on desiccation tolerance, their ability to maintain water within their system. And by coupling these two studies, it'll ultimately reveal how ecological and sexual selection pressures interact with the system. And so by coupling different methods from genetics and animal behavior and applying these methods to the housefly system, we can hopefully see how genetics and the environment interact to regulate interesting behaviors. And then we can look at the chemical ecology of this system to ultimately see how a trait, such as cuticular hydrocarbon profile, evolves when it's under both strong sexual as well as ecological selection. Well, there's a whole army of students and collaborators I want to thank. I'll just leave them, leave their names up here for the sake of time and take any questions if there's still time. And thank you.
Thank you very much, Pablo. We do have some time for questions. If people have questions,
sure. I'll stop sharing my screen.